Well, I'd like to thank Vanessa for the invitation and for arranging this superb Congress and for the opportunity to speak. This is a challenging topic, but I hope I can cast a little light on it for you. So what I'd really like to do is divide the talk into three parts. First of all, I want to begin by discussing what the term intellectual disability means, or what its current definition is. I'd like to talk about what we know about the anatomy and physiology of intellectual disability, and then finish with some comments about intellectual disability in CDG, and Dr. Serrano's beautiful talk is a very nice introduction to that. So intellectual disability is not just a number, and I think that's a very important point, and again, Dr. Serrano, I think, made that quite clear. This is the definition that is used by the American Association for Intellectual and Other Developmental Disabilities, and I've taken this from their website, and this is a great resource that I'd encourage you all to use. This is a large, very well-funded organization that supports two separate journals, so it's a great resource, I think, for this community. But they define intellectual disability as a symptom complex that's characterized by limitations in both intellectual functioning and adaptive behavior. So again, it's not just the number, it's the fact, how does this cognitive difficulty cause problems in everyday life, in social skills and practical skills? And it must originate before the age of 18. So in other words, this is a developmental disorder, and this helps us differentiate intellectual disability from dementia. Now, dementias can start in children, but intellectual disability is the type of cognitive problem which is most common in the CDGs, which are essentially developmental disorders to a very large extent. Well, what do we mean by intellectual functioning? And this really refers to general mental capacity. So it's our ability to learn, to reason, to solve problems, and so on. It's the broad sweep of things that humans can do in general better than other animals. Other animals can do almost everything better than us in certain areas, but we have a bigger neocortex, and we're usually better at reasoning. Not always. Not always. We have lots of politicians who disprove that, but we're usually better. And we need a number. And the point that Mercedes made in the previous talk was that when we're assessing people, we need to be able to generate numbers as a means of assessing their progress, and when we treat, to assess the value of that treatment. So the standard treatments are intelligence quotient tests, and you've heard the names of some of these. We could spend the whole day talking about different tests. The important thing is, if you're going to use a test or an instrument, which is the term that's often used, it should be something that has been developed for a specific population and validated. In other words, you've developed a theoretical framework, then developed a tool, and then you try it in the population. And again, just as Dr. Serrano said, you may need to make certain adaptations. So if you're going to use a well-known instrument such as the Wechsler Intelligence Scale for Children, or the WISC, which is now in its 5th revision, you may need to give people more time than standard if they have physical or hearing or other processing problems, which would give a falsely low result. And of course, you can't, for example, administer a test that's been developed in English to someone who speaks Spanish. That sounds so obvious, but sometimes people forget these things. So using with all of those caveats, if you use a standard test such as the WISC in children, if you have a full-scale intelligence quotient of 70 to 75, that indicates that there are some problems with intellectual functioning. But you also want to know how well people can adapt to these limitations. And again, adaptive behavior is defined as conceptual, social, and practical skills that are learned and performed by people in their everyday lives. Now, there may be people whose IQ might be 70 or 75, but they may actually have certain abilities, for example, in animal husbandry, take an example, such that they're really highly skilled in those areas, and that in their day-to-day activity, having that IQ of 70 to 75 doesn't limit them significantly. So such a person would not be said to have an intellectual disability. And I think that's a very important point to bear in mind. We have to make sure we capture people's strengths as well as their areas of relative weakness when we assess this. And when we think about adaptive behavior, again, we can further subdivide this, so we can think about conceptual skills, language and literacy, the ability to manage money, time and number concepts, and also self-direction. So can people organize themselves? And then we come to social skills. And again, How do people interact on an interpersonal level? Do they understand social responsibility? Do they have self-esteem? And of course, for many children and young adults, the issues of gullibility and naivety. So these people are vulnerable because they may not understand the concepts that not everybody is good. They need to be able to solve social problems, to follow rules, and to understand how they apply them, and again, to avoid being victimized because of their difficulties with these concepts. And of course, there are practical skills. Can people take care of themselves? Can they dress and undress? Can they prepare meals? What occupational skills do they have? Do they have access to health care? Do their intellectual problems cause difficulties with that access? How do they do with travel and transportation? Someone may not be able to get a license to drive, for example. Can they manage public transport? Do they need assistance with that? Can they follow schedules and routines? Are they safe? Do they understand how to use money? How to use the telephone, for example? All very basic things that we must take into account in assessing somebody's intellectual capacity. And of course, there are a number of additional factors that set this in a framework. So what is the community environment? What sort of social setting does a patient come from? Again, some communities are much more supportive than others, and sadly, very highly developed Western cultures place a great value on intellectual capacity, and so people who have limitations in those areas may paradoxically be worse off than in communities that place less excessive value on that. We have to understand, of course, there's great diversity, not only in language, but in the way cultures communicate with one another, how they move and behave. All of these things have to be taken into account in assessing intellectual capacity. And again, I can't overemphasize the fact that sometimes we tend to overemphasize limitations. Let's look at the strengths that people have. As I mentioned, some people can have so-called savant skills, although their general level of functioning may be in a range that suggests limitations. They may have particular skills which can be tremendously valuable to them and allow them to adapt. And I think one of the things, again, that's been alluded to is the fact that the way that you function in life will improve if the support that's provided to you is tailored and provided over a sustained period. And I can tell you, in practicing in the United States, one of the great problems we have is that when we're trying to get therapies to support patients, the models which are used by insurance companies are all based on adults who've had strokes, which has very little to do with children and young adults who have an ongoing problem. The problem doesn't go away after 20 sessions of therapy or 6 months. So we often have to advocate very strongly for that. So that's a background, and I suspect I didn't say anything that you're not all very much familiar with from your own experience. What do we know about the neurological bases, though, of intellectual disability? And this is a challenging area for us as neurologists because, as you may know, neurology really became a separate specialty in the 19th century. And in fact, it's interesting that we're here in France today because localization is one of the keys to neurology, and that is understanding that injury to a specific part of the brain will cause specific neurologic symptoms and signs. And really, many people would say it was Broca in this country 150 years ago in 1865 who first recognized that very specific injury to the inferior frontal gyrus of the brain causes the inability to express thoughts in the form of language, so-called expressive aphasia. And this has become a mantra and something that we do. I used to, when we had oral examinations for neurology boards in the United States, the first things we would ask our candidates to do was to say, where do you localize this neurological problem? So this is a standard thing we do. But what happens when we deal with intellectual disability? We can't do that. The vast majority of patients, we can't say, well, here is the part of the brain that's responsible for intelligence. And we know that people can suffer quite severe localized injuries, but their overall intelligence is preserved. So we have to think about this type of disorder, as with other disorders such as autism, in a different way than we do in classical neurology. And what has emerged over recent years is that there are multiple networks. We have to think of neurons working together in networks, not just in one part of the brain, but it's how these neurons interact with each other, how they integrate information. Now, that's not a new concept. Sir Charles Sherrington won the Nobel Prize in the early 20th century for his work in explaining how the nervous system integrates information. He wrote a famous text about this in 1906. So what are the factors that contribute to developing these networks? Well, first of all, you have to make neurons. the process of neurogenesis. And this occurs well before birth. This is beginning really becoming quite traumatic in the middle trimester of pregnancy. We have many more neurons than we need before birth. In fact, the normal developmental process involves pruning back neurons, losing neurons in a programmed fashion. If they don't make the right connections, they won't survive. So this is an important process. Neurons, of course, originate around the central fluid-filled cavities in the brain, the ventricles, and then they migrate in waves to the surface of the brain. An immensely complex process that occurs mainly during the middle and final trimester of pregnancy, but actually occurs to some extent in postnatal life. It's amazing to me that anybody has a normal brain. It's so complicated, and yet in most cases it works well. But if it doesn't work, you will not have normal circuitry, and that can cause intellectual disability. It may cause seizures and other problems. And one of the other important factors is getting down to a molecular level that cells have to signal to one another to develop appropriately. And if these so-called signaling cascades are disrupted, again, one of the consequences may be widespread dysfunction of neuronal networks and intellectual disability. And then we get down to some very basic mechanisms in neurology. As you all know, all of the molecules all of the structures in our body, all of the proteins are encoded by our DNA. This DNA has to be transcribed into messenger RNA, which is then translated into protein, and the proteins are then modified, and glycosylation is one of the major modifiers of proteins. But abnormalities in the regulation of this transcription and translation process can also give rise to intellectual disability, and of course problems with connections between neurons. And so that implies problems before the synapse, the gaps between the neurons, problems with the formation of vesicles for neurotransmitters, which are then released in the synapse, the gap between neurons, problems with the synapse itself, its plasticity may be abnormal, the dendrites, which are the projections from neurons which receive information, may be abnormal, and of course there may be problems in what's called the postsynaptic region. Okay. Let me just show a couple of illustrations because I deluge you with a lot of information, and if you're not a neurologist, this may have all seemed like gobbledygook, and I apologize. So now I'm going to show some images to make it a little bit clearer. And again, some basics of brain structure. We have the cerebral cortex, the cerebrum here, the cerebellum, which we've heard described so eloquently, and we've also understood from what Dr. Serrano presented to us that it's not just involved with balance and coordination, it has very important functions in intellectual ability and in language. We can divide the brain into lobes. The frontal lobe, the largest lobe of the brain, the parietal lobe, which is involved with multisensory integration, the occipital lobe with vision, the temporal lobe with language and memory. The frontal lobe is a sort of governor for the brain. It inhibits a lot of other activities. It's important in planning. But having said all of that, those localizations don't explain everything that's going on. It's the integration of activity that's important. And of course, we have a lot of basic functions occurring deep in the brain, in the diencephalon, the hypothalamus, the brain stem here, and in the spinal cord, which disseminates this outflow of information to the rest of the body. And there are these structures you may have heard of called the basal ganglia. This is what they look like in 3 dimensions. These are structures deep in the brain which are very important relay nuclei which receive information from the cortex, from the internal parts of the body, from external sources, and integrate it to modulate our behavior and movement and language and so on. They're very complex. We don't understand them well. Again, classical neurology is what we might call the neurology of deficit. So we understand to a reasonable extent what happens if we damage these structures. So, for example, if the caudate nucleus, whose head is shown here, is damaged, one of the diseases that can result is Huntington's disease, you may have heard of. Other types of movement disorders can occur with damage to different parts of the basal ganglia. But really we're talking more about what's going on at the very basic level of the neuron, which has a body containing the nucleus. These dendrites which receive information from other neurons, it's transmitted through the axon and then disseminated through axon terminals, which terminate either on other neurons or, in the case of motor neurons, on motor fibers. And one of the ways in which our nervous system is able to be more efficient is because these nerve fibers are insulated with a substance called myelin. And you may have heard about this. Myelin myelination is something that occurs mainly during postnatal life. One of the questions I like to ask my residents is, why is it when you examine a newborn baby who has virtually all the structures in the brain, why can't this baby talk to you? Why can't the baby get up and walk and sing and tap dance and do other interesting things? It's because the nerve fibers lack myelin. And so the brain can't integrate information normally. And this process of myelination correlates very well with developmental stages that we recognize in children. And so if this is disrupted, that can have multiple consequences as well. And we know in some CDGs it may be abnormal. And this is just a little cartoon again showing a cell called an oligodendrocyte in the central nervous system, which extends processes which wrap around the axons of nerve fibers and result in myelination. Okay, having said all that, recent studies, and there's been a huge progress in all of medicine, particularly in neurology, because of the genetic revolution. We can now sequence DNA and discover new genes much more readily than was the case 20 or 30 years ago. And so there are at least three common pathways we see in intellectual disability. The first is in the synapses. These are the gaps between nerve cells where the information is conveyed not by electrical impulses but by chemical transmitters. And there are at least 50 of these. The number keeps increasing as we understand better. So again, the details of these particular synapses and neurotransmitters and receptors are not important, except to say we know with all of those that I've listed, mutations can cause intellectual disability with or without any other symptoms. Similarly, I mentioned before that there are these cascades in intracellular signaling, which I'll explain a little more in a moment. Disruption of these produces a number of syndromes in which intellectual disability is a major feature. And finally, the ability to turn on and off transcription, in particular, and translation to a certain extent, can produce a number of disorders. These are very basic cellular functions, including intellectual disability. Again, for those of you who may not be so familiar with this, the synapse is the gap between neurons. A man called Ramon y Cajal over 100 years ago won one of the first Nobel Prizes for showing from very careful studies of the nervous system that there are actually gaps between nerve cells. One of his intellectual opponents, a man called Camillo Golgi, with whom he shared the Nobel Prize, thought that the nervous system was just a network with no gaps. But these are important because the neuron needs to manufacture neurotransmitters. It concentrates them in these little vesicles, which, when an electrical signal reaches the end of the nerve cell, cause release of the neurotransmitters. They have to be detected by receptors in the postsynaptic membrane. And as you can imagine, things can go wrong at every level here. And if they go wrong, it can produce a number of problems, including seizures, intellectual disability, weakness if it occurs at the gap between nerve cells and muscle, and so on. So one of the major excitatory neurotransmitters in the nervous system is called glutamic acid. It has what are called metabotropic and ionotropic receptors. Again, the details are not important, but just to make the point, there are multiple receptors that can detect these neurotransmitters. There can be mutations that affect the function of all of these receptors, and the result of that can be intellectual disability. Here's one of the signaling cascades. It's called the RAS-MAPK pathway. Again, the details are not important, except to point out that there are very fundamental cellular processes that depend on these signaling pathways being intact. And in fact, this slide shows you some of the disorders that can be associated with abnormalities in this pathway. Again, many of you may have heard of some of these syndromes, the Noonan syndrome, neurofibromatosis, a neurofibromatosis-like syndrome, a family of disorders called the cardiofaciocutaneous syndromes, the Leopard syndrome, and so on. The point to be made is, again, these are very basic cellular processes. For reasons we don't understand, a mutation in the genes encoding any one of these particular molecules can cause a very specific syndrome. And again, this just shows the relationship between the RAS pathway and what's called the P13K mTOR pathway. Again, basically the RAS pathway is involved in cells proliferating. The mTOR pathway is involved in keeping them alive once they've developed. And again, a whole bunch of syndromes associated with disruption of this pathway, the tuberous sclerosis complex, which is a fairly frequent cause of intellectual disability, together with other symptoms. The P10 gene can be mutated not just in glioblastoma, but in a variety of overgrowth syndromes as well. Again, the details are not important, but just to recognize these are very basic pathways, all of which can be affected by glycosylation because they all rely on proteins. Almost all of these proteins are glycosylated. If they're malfunctioning, it gives a potential explanation for intellectual disability in congenital disorders of glycosylation. And finally, we mentioned gene expression. Genes, of course, are sequences of bases on deoxyribose nucleic acid, on DNA. That's transcribed into messenger RNA, which is then processed. It passes out of the nucleus into the cytosol in the cell, and on the ribosomes, it's translated into proteins. And as we mentioned, one of the most important modifications of these proteins is glycosylation. But there can be abnormalities at all of these levels, which again can produce highly specific syndromes. Intellectual disability is a common feature of all of these. Again, this is just a little cartoon showing how RNA, histones, and methylation of DNA can all interact to alter the way that protein in general is being expressed in cells. And there's a whole bunch of diseases now that we recognize. Again, these all feature intellectual disability. It's not exhaustive. You've probably heard of Fragile X syndrome, perhaps the Angelman and Prader-Willi syndromes, Rett disorders. All of these are disorders of the regulation of transcription of DNA. So, having given you all of that information, what does it mean for CDG? Well, I think the important thing is that intellectual disability, as you know, is common in CDG, but it's not universal. And even within a particular subtype of CDG, as you've heard, it's not universal. So we know that this is a result of developmental expression of these diseases. There must be a dosage effect. And in general, with disorders such as the CDGs that result from deficiencies of enzymes, as a general rule, the more enzyme activity you have, the less severely affected you are. Now, there are multiple other factors that interact because, of course, it's not just that one gene that determines how you turn out. You've got 20, 25,000 others that are interacting as well. But it is important that it's developmental, it's dose-related, and there are many types of CDG where we can't clearly look at the anatomy of the brain and say, here is the explanation for the intellectual problems these patients have. We particularly think of the TUSC3 form of CDG, where patients may have no other manifestations of CDG other than intellectual disability. We do know in some cases there are migrational disorders. They're quite common in the O-linked disorders of glycosylation, such as the congenital muscular dystrophies, where the lack of normal barriers causes neurons to actually migrate through the surface of the brain, producing a syndrome called cobblestone lyssencephaly. So we have many clues, but not a large number of answers. Well, what can we do for patients with intellectual disability? And again, you've heard beautifully from Dr. Serrano how early intervention is important, that we need to understand and anticipate the needs that children may have. We have to make sure that they receive appropriate education, but we also have to educate other people, those who interact with the children, so that they can have appropriate expectations so that children can succeed to the best of their ability. And of course, as life goes along, we have to think about employment possibilities to ensure that these children have access to healthcare, which can be a challenge for patients with rare diseases, as you all know, to think about preventing complications of these diseases. Glycosylation occurs, of course, throughout life, but it's very prominent in early development. So if we're going to affect the outcome, we need to make the diagnosis early and intervene as early as possible to be maximally effective. And there are a whole bunch of legal issues we have to take into account, particularly as children go older, in terms of their vulnerability, guardianship, protecting them, and making sure that their rights are achieved. Okay, well that was a great deal of information. I hope it wasn't too overwhelming, but I'm excited about understanding these disorders. I'm very excited about being here to meet with this group again and to meet with the true experts who are the people sitting in the room here. Because you live with these disorders 24 hours a day, 7 days a week, 365 days a year. So you are the true experts. I like the opportunity to sit down and talk to people and learn from you. I'm happy to answer any questions. And again, thank you for being here.